Hello guys, this is Jeong Kang from Sweet Lab. Our lab is Sustainable Water Environment and Energy Technology. Our lab is researching on practical environmental improvement, such as improving water quality and generating energy from waste. I'm going to introduce about my first research area, removal of contaminants by applying electricity called as Electro Oxidation. Recently, lots of researchers found that there are so many nanosizes of toxic compounds in clean-looking water because these compounds cannot be removed by conventional water treatment system. Also, there is high contaminated water from recycling water system. Electro Oxidation is one of the technologies that cleaning the kinds of water. Very strong oxidant, hydroxyl radical, can be generated by applying potential. And with this radical, toxic compounds can be degraded to carbon dioxide and other minerals. Our lab is working to not only increase the removal efficiency, but also decrease the energy consumption by developing this kind of membrane type electrode. Because toxic compounds can be removed more efficiently by passing through so many nanosize of pore on membrane electrode. Also, we can obtain directly the clean water by membrane separation. Now I'm gonna show the system briefly. When put this electrode on beakers, and when applied the voltage, contaminant can be removed. After 1 hour, we can get clean water like this, as you can see the color change like this solution. It was the brief introduction about my first research area. Now I briefly tell you about my lab while we move on to the fourth floor. Our lab consists of 3 postdoctors, 8 of PhD students, 9 of master students, total 20 of lab members. Three or four of them make a research team and get lots of meaningful achievement. Also, we have so many analysis instruments. So we can do experiment freely and discuss the result creatively. So we arrive on the fourth floor. From now on, let's move on to the second research area, membrane bioreactor.
Hi everyone, I'm Kiki. I'm a PhD student from Sweet Lab. Today I want to briefly explain about you guys about my reactor and my research. So follow me. This is my reactor. This one I use the biological technique method to treat the wastewater. So the wastewater was pumped to the first tank, which is an anaerobic tank, and in this tank, some microbial cell will change the form of phosphorus. And the second one is anoxic tank. In here, the denitrification process will occur and produce some nitrogen gas as a product. So nitrogen will remove from the wastewater in anoxic tanks. And the last one is aerobic tank. In this tank, I also put the membrane here to separate the to separate the treated water from the microbial cells. So when you using this kind of reactor, that's called A2O MBR. A is come from anaerobic and anoxic, and O is from oxic, and MBR is stand for membrane bioreactor. So when you use this kind of process, you can treat both nitrogen and phosphorus, which is the main contaminant in the wastewater, and also it's cost effective and easy to operation. And the most important thing is it environmental friendly process because no chemical need to treat the contaminant in wastewater. And this is my reactor and my research. So the next person will explain you guys about how to recover the energy from the waste.
Hi, my name is Hyung-Seung Ham. I'm a second-year master's student researching on energy recovery from organic waste. I'll show you briefly what I do in the Sweet Lab. So, in a food waste like this, is full of energy-rich macronutrients, such as carbohydrates, lipids, or protein. And just like how we digest food and convert them to energy, so does the microorganisms. So when we add these food waste into a anaerobically cultivated consortia of microorganisms, they break down these macronutrients into tiny little monomers, then to organic acid, and finally produces gas. And this process is called anaerobic digestion. So the gas that these microorganisms produce are full of energy-rich methane. And we can use this methane to convert into electricity or just use it like a natural gas. By using this gas chromatography machine, we can analyze how much methane content is inside the biogas and ultimately can analyze what parameters affects this methane content. Among these consortia of microorganisms, some of them utilizes a very special mechanism of electron transfer between different species. So by adding these lab-made conductive material inside of a bioreactor, we can expedite the anaerobic digestion process and therefore improve the efficiency of the reactor. So I talked briefly about what anaerobic digestion research I do. The next is an exciting research of improving water quality by using 3D printer.
Good afternoon, everyone. I'm Jisun Lee, and I'm master student in Sweet Lab. Today, I'm going to introduce 3D printing technology that our lab is working on. By using 3D printing technology, we can produce almost every stuff that we design. As you can see here in the software, we can design the 3D objects and the software slices layer by layer like this. Now we can get the data and let's move on to the 3D printer. When we put the data into the 3D printer, now we can print what we designed. This 3D printer is conducted by digital light processing, and these are the 3D objects that our lab prints for the research. The first one is an electrode made of carbon nanotube, which is conductive membrane electrodes, and we are trying to design diverse structures. This is inline mixer. In order to increase solubility of ozone gas in the water, we designed this kind of complex inner structure by using 3D printer. This is ceramic membrane, and after printing this, by using this membrane reactor, we operate membrane and oxidation hybrid process. This is the end of presentation, and let's move on to the Q&A session. Thank you.